There is something inside you that is older than you are. Not a feeling, not an instinct in the loose, casual sense of the word. Something encoded, stored, passed down through a chain of bodies, stretching back further than any written record can follow. Scientists have a name for the mechanism. Several names, actually, depending on which layer of the biology they are describing. What they do not have is a complete explanation for what it means. Why do some studies suggest that stress, famine, and trauma may leave biological traces beyond a single lifetime? Why does the body of a grandchild show measurable chemical differences that correlate with a catastrophe the grandchild never lived through? Why does a stretch of DNA carry markers that some researchers believe were shaped by events that ended generations before you were born? You will hear the phrase genetic memory used tonight. It is worth saying clearly, at the start, that this phrase is not a scientific term. It is a shorthand, a way of pointing towards something real that the science is still working to define precisely. What the researchers have found is more specific, more careful, and in some ways, more quietly astonishing than the phrase suggests. Tonight, we are going to follow the actual findings rather than the shorthand. Not to give you easy answers. The science here is genuinely unfinished, but to stand at the edge of what has been found, carefully and honestly, and look at something that may change how you understand what you carry. Settle in. Let your breathing slow. Think of the women who were pregnant in a city under siege in the winter of 1944, eating tulip bulbs to survive, not knowing if the spring would come. Think of the children they carried. Think of what those children's cells may still hold. Decades later, long after the siege ended and the city rebuilt and the world moved on, that is where this story is going. And it is quieter, and stranger, and more human than the phrase genetic memory suggests. This story may have left traces that no one expected to find. Chapter 1. The body that responds without being taught. Start with something ordinary. You are walking alone at night. A quiet street. Familiar enough. Nothing unusual about it. And then something shifts. Not a sound you can identify. Not a shape you can name. Just a change in the quality of the darkness ahead of you. A slight movement at the edge of your vision. And your body responds before your mind has finished forming a single thought. Your heart rate climbs. Your breath shortens. The muscles along your shoulders pull inward. Your eyes widen and your attention narrows to a point, cutting out everything except the thing that moved. You process all of this afterward, with your thinking mind, and you tell yourself a story about what happened. You were startled. You were tired. The neighborhood felt unsafe. But the response itself, that immediate, total, full-body recalibration, happened before any of those explanations existed. It happened in the space between perception and thought, in a place your conscious mind does not govern and cannot fully access. Most people explain this as instinct, and for most purposes, that explanation is enough. But there is a different kind of response, harder to notice, harder to explain, and it is the one this story is actually about. Some people move through the world carrying a heaviness they cannot trace to any experience in their own lives, a baseline alertness that never fully relaxes. Even in genuinely safe situations, a specific sensitivity to certain kinds of threat, certain kinds of loss, certain kinds of separation, that seems disproportionate to anything their personal history would account for. A body that behaves as if it learned something the person themselves was never taught. Therapists encounter this regularly. Not as mysticism, as a clinical pattern that resists the usual explanations. The person sitting across from them did not experience the trauma their nervous system seems organized around. They grew up in safety. They were not abused, not neglected, not exposed to the specific events their body appears to be bracing against. And yet, The body braces. For a long time, the most available explanation was psychological. Learned behavior. Subtle transmission of anxiety through parenting. The way a frightened parent holds a child. The way certain silences in a household carry weight. The unspoken rules of a family organized around a wound nobody talks about directly. These are real mechanisms. They are well documented. They explain a great deal. But they do not explain everything. Because in some cases, the researchers who started looking more carefully found something that the psychological explanation alone could not account for. They found it not in behavior, not in the patterns of how families communicated across generations, but in biology, in the actual molecular structure of cells, in the way certain genes were being expressed or suppressed, or held in a particular configuration that the person's own life experience did not seem sufficient to produce. This is where epigenetics enters the story. The word itself is less intimidating than it sounds. Genetics is the study of genes, the sequences of DNA that carry instructions for building and running a living body. Epigenetics, from the Greek prefix meaning above or upon, is the study of changes in how those genes behave without any change to the underlying sequence itself. Think of it this way. Your DNA is a very long text. It contains instructions for everything your cells know how to do. Epigenetic changes are not edits to that text. They are annotations, marks placed on top of the text that tell the cell to read this section more actively, or more quietly, or to set it aside for now. The text stays the same. What changes is how it is being read. Those annotations can be influenced by experience, by stress, by nutrition, by the chemical environment a body moves through over a lifetime. And the question that researchers began asking, carefully and with considerable disagreement among themselves, is whether some of those annotations can be passed from one generation to the next. Not the experiences themselves, not memories in any conscious sense, but something more subtle and in some ways more unsettling. A biological configuration, a set of adjustments to how genes are read, shaped by what a parent or grandparent survived, that may influence the biology of someone who never lived through any of it. The research exploring this possibility is real. It is published in peer-reviewed journals. It involves serious scientists at respected institutions. It is also contested, debated, and far from settled. The honest position, and this story will hold that position throughout, is that the evidence points towards something genuinely interesting while stopping well short of the dramatic conclusions that popular accounts tend to reach for. What the evidence does suggest, within those limits, is this: the body may keep a certain kind of biological record, not a narrative record, not a memory in the way that word is usually meant, a molecular record, a set of adjustments that carry information about the environments in which previous generations survived. And in some circumstances, under conditions, researchers are still working to define precisely. Traces of that record may persist beyond a single lifetime. That is a careful claim. It is also, if you sit with it quietly for a moment, a remarkable one, because it means that the body you are moving through the world in right now may not be only the product of your own experience. It may carry, in ways that are still being measured and still being understood, something of the biological history of the people who made you possible. The science is not certain about the full scope of this. The researchers working on it will tell you that themselves, and the ones who do not tell you that are the ones worth being most skeptical of. The observation that started this line of inquiry was simple enough to describe, specific enough to measure, surprising enough that it could not be easily set aside. It began with a smell, and with mice that were afraid of something they had never encountered, whose children carried the same response in their nervous systems, expressed in the same measurable biological patterns, for reasons that their own lives gave them no basis for. That is where the next part of this story begins. Chapter 2. What epigenetics actually found. Begin with a mouse and a smell. In 2013, a team of researchers at Emory University in Atlanta published a study in the journal Nature Neuroscience that would be cited, debated, and argued over for the years that followed. The researchers were Brian Dias and Kerry Ressler. What they did was straightforward enough to describe in a few sentences. They took a group of male mice and exposed them repeatedly to a chemical called acetophenone, which smells faintly of cherry blossoms. Each time the mice encountered the smell, they received a mild electric shock. The mice learned quickly to associate the scent with something unpleasant. Their bodies responded to it the way bodies respond to threat. Then Dias and Ressler let those mice father offspring. The offspring had never smelled acetophenone. They had never received a shock. And yet, when researchers introduced the scent to them, they showed a heightened sensitivity to it. A stronger response than mice, conditioned to other smells. A nervous system that behaved measurably, as if it had already been introduced to this particular threat. The effect appeared in the following generation as well. When the researchers examined the biology more closely, they found structural differences in the part of the brain dedicated to processing smell. The mice whose ancestors had been conditioned to fear acetophenone had more receptor cells dedicated to detecting that specific molecule. Their brains had been physically organized around a threat their own lives had never presented to them. And in the sperm cells of the conditioned fathers, the researchers found epigenetic marks on the gene responsible for producing the acetophenone receptor. The annotation on the text had changed, and that annotation appeared to have been passed along. Set that finding down for a moment and breathe with it. A father learned to fear a smell. His children arrived in the world already leaning toward that same fear. In their biology, before their first breath, that is what the study showed. Now consider what it did not show. Because the gap between those two things is exactly where honest understanding lives. What Dias and Ressler demonstrated is that a specific conditioned fear response produced measurable epigenetic changes in sperm cells that correlated with altered nervous system development in offspring. The effect was real. The methodology passed peer review at a rigorous journal. What the study did not demonstrate is that this mechanism operates in humans in the same way, at the same scale, or with the same reliability. Mice and humans share a great deal of biology. They are not the same organism. The study also faced replication challenges in the years that followed. Other researchers attempting to reproduce the results found the effect difficult to replicate consistently. Kerry Ressler himself, in subsequent work and interviews, described the findings as suggestive rather than conclusive, as opening a question rather than answering one. That is the honest scientific position. It deserves to be held as such. To understand why transgenerational epigenetic inheritance is such a contested idea, it helps to understand the model it challenges. For most of the 20th century, the dominant view in biology held that information flows in one direction. From DNA to the organism, the environment could shape the individual during their lifetime. But those environmental influences could not be written back into the genetic material and passed to offspring. Each generation started fresh. This model was elegant. It was also, researchers began to discover from the 1980s onward, incomplete. The epigenetic marks were real. Their responsiveness to environment was real. The question was whether they survived the process of reproduction. When sperm and egg cells form, and again, during the earliest stages of embryonic development, the epigenome undergoes a nearly complete erasure and resetting. The system appeared designed to prevent the transmission of parental experience to offspring. But not all marks were erased. Some, in some locations, in some organisms, under some conditions, appeared to escape the reset. That sentence is worth reading slowly. Not all marks. Some, In some locations, in some organisms, under some conditions, that is the actual scope of the evidence at this stage of the story. Real enough to take seriously, limited enough to require precision about what it does and does not imply. The mouse study offered a specific, measurable example of a mark that seemed to escape. It was not proof that the phenomenon was widespread or robust in mammals. It was evidence that the phenomenon could occur under specific conditions, in a specific organism. That is a meaningful finding. It is also a much more limited one than the headlines it generated. Understanding this gap between what a study shows and what it is reported to show is essential for following this subject honestly, because the gap appears again and again in the epigenetics literature, and closing it is the difference between understanding what the science has actually found and carrying away a story that sounds satisfying but does not accurately reflect where the evidence actually stands. The evidence, at this point in the story, supports the following. Epigenetic marks exist and influence gene expression in real and significant ways. Some marks respond to environmental conditions. In some organisms, particularly simpler ones, some marks can survive the reproductive process and influence the biology of offspring. In Miche, the evidence suggests this can happen under specific conditions, though replication has been inconsistent. In humans, the question is open and actively being investigated. That is the foundation, solid in its lower levels, less certain as it rises. What researchers found next pushed the question out of the controlled world of laboratory mice and into the vastly more complicated territory of human history. And history, as this story will keep demonstrating, is where the most difficult and most important questions tend to live. Chapter 3. The Famine That Left Marks on Grandchildren. There is a village in northern Sweden called Överkalix. It sits close to the Arctic Circle, in a county called Norrbotten, far enough from the centers of European history that the catastrophes which periodically reorganized the rest of the continent largely passed it by. What Överkalix did experience, with regularity and severity, were the consequences of its own geography. Short growing seasons, long winters, harvests that could fail without warning and leave a community facing months of genuine hunger before spring returned. And, because Överkalix was isolated enough that its population records were kept with unusual consistency across several centuries, it became, entirely without intending to, one of the most important data sets in the history of epigenetics research. In the 1990s, a Swedish researcher named Lars-Olof Bygren began examining those records with a specific question in mind. He wanted to know whether the nutritional environment experienced during a particular window of childhood development had measurable effects not just on the individual who experienced it, but on their descendants. The window he focused on was a narrow period in early adolescence, roughly between the ages of 9 and 12. When the reproductive system is maturing and appears to be particularly sensitive to environmental signals, researchers call this the slow growth period. Bygren could trace, for specific individuals in the Överkalix records, what the harvest conditions had been during those years of their development. He could then trace the health outcomes of their children and grandchildren, following the lines of a community's life across 3 generations of careful record-keeping. What he and his colleagues found was a statistical pattern that conventional genetics alone struggled to explain. Men who had experienced food abundance during that developmental window had grandsons who showed higher rates of cardiovascular and metabolic disease. Men who had experienced food scarcity during the same period had grandsons with lower rates of those conditions. The nutritional experience of the grandfather appeared to echo, measurably, in the health of the grandson. The Överkalix data is epidemiological. It is a pattern observed in historical records, not a controlled experiment. The sample sizes were modest, Confounding variables could not be fully accounted for. Icahn and his colleagues were careful about this in their published work. The pattern was real and statistically significant. The mechanism behind it was a matter for further investigation. Not a conclusion the data alone could support, but it pointed somewhere. And it sent researchers looking for a population where the disruption had been severe enough, and the medical records detailed enough, to examine the biology directly, rather than inferring it from century-old harvest data. They found it in the Netherlands. In the autumn of 1944, the German occupation authorities cut off food supplies to the Western Netherlands in retaliation for a Dutch railway strike called in support of the Allied advance. What followed was one of the most severe famines in modern European history. The Dutch called it the Hunger Winter. The Hunger Winter. Between November of 1944 and the liberation of the Netherlands, in May of 1945, roughly 4.5 million people in the Western Netherlands survived on rations that fell, at the worst point of that winter, to below 400 calories per day. The normal adult requirement is roughly 2,000. People boiled tulip bulbs and ate them. They burned their furniture for warmth. They watched their neighbors grow thin, and then thinner, and then sometimes not recover. The death toll from starvation and cold reached approximately 22,000 people before the spring came. Among those who survived were women who were pregnant during it. Their children, born into a liberated and gradually recovering Netherlands in the months that followed, grew up to become one of the most studied populations in the history of epigenetics research. Because the famine had a clear start date and a clear end date, and because Dutch medical record-keeping was meticulous, researchers could identify specific individuals who had been exposed to severe undernutrition during specific trimesters of fetal development. They could compare the health outcomes of those individuals across their entire lives, with people born just before or just after the famine period. What the research found, accumulated across decades of follow-up studies, was a pattern of lasting biological differences in the famine-exposed generation. People who had been in the womb during the worst of the hunger winter showed higher rates of obesity, cardiovascular disease, and metabolic disorders in adulthood. They showed differences in glucose metabolism and stress hormone regulation that persisted for decades after the famine itself had ended. And when researchers examined their DNA, they found something specific. At a region of the genome associated with a gene called IGF-2, which plays a role in fetal growth and metabolism. Famine-exposed individuals showed measurably lower levels of methylation compared to their siblings conceived after the famine ended. The epigenetic mark was different. The annotation on the text had been altered by the nutritional environment of the womb, and that alteration was still detectable 60 years later. Think about what that means for a moment. A woman stood in a city without food in the winter of 1944. She was cold and frightened and hungry in a way that most people reading this have never been hungry. Inside her body, a child was forming, and the environment of scarcity that surrounded them both left a mark in that child's cells. A specific molecular adjustment to a specific gene that researchers could still measure when that child was in their 60s. Living in a peaceful and prosperous country that bore almost no resemblance to the city their cells had learned to survive in. The famine was real. The mark was real. The distance between them, across 60 years of ordinary life, is what makes the finding remarkable. This is the most robust finding from the Dutch hunger winter research. The methylation difference at IGF-2 in the directly exposed generation is well supported by multiple studies using rigorous methods. It is not seriously contested. The next question is where the research becomes more contested. Some studies have reported measurable differences in the children of famine-exposed individuals. A study, published in 2015, found evidence of some altered methylation patterns in the children of hunger winter survivors, particularly those whose mothers had been exposed during early pregnancy. These findings are real. They appear in peer-reviewed literature. They have been taken seriously by researchers in the field. But they are not as robust as the findings in the directly exposed generation. The sample sizes are smaller. The effect sizes are more modest. The confounding variables are harder to account for. Several researchers working carefully in this area have noted that the evidence for effects in the grandchildren is suggestive at best and should not be presented as established. The honest picture of the Dutch hunger winter research looks like this. In the generation directly exposed to famine in the womb, the evidence for lasting epigenetic change is strong. In the next generation, the evidence for some biological transmission is present and worth investigating further. Beyond that, the evidence thins to the point where confident claims are not warranted. That graduated picture is less dramatic than the version that circulates in popular accounts, but it is the accurate one. And even in its accurate form, it asks a question that does not go away. A catastrophe ends. The visible world recovers. The food returns. The cities rebuild. A child grows up in peace, speaking a language their grandparents spoke, living in a country that has done its best to move forward. And, somewhere in their cells, at a specific location, on a specific gene, a molecular adjustment persists. Not a memory, not a story, a mark, left by a winter that ended before they were born. Chapter 4. Trauma's Biological Signature. There is a particular kind of research that requires its subjects to revisit the worst thing that ever happened to them. Not once, repeatedly, in clinical settings, with fluorescent lights and blood pressure cuffs and researchers taking careful notes, while the person sitting across the table describes what they saw, what they lost, what they have never fully been able to put down. Rachel Yehuda has been doing that research for more than 30 years. Yehuda is a professor of psychiatry and neuroscience at the Icahn School of Medicine at Mount Sinai in New York. She began her career studying post-traumatic stress disorder in Vietnam veterans, men who had come home from a war and found that the war had not entirely come home from them. Over time, her work moved toward a question that the standard clinical model of trauma had not adequately addressed. She wanted to know why some people exposed to severe trauma developed lasting physiological changes, while others, exposed to comparable events, did not. And she wanted to know whether those physiological changes could be associated with measurable biological differences in people who had never experienced the original trauma directly. Her most discussed work involves Holocaust survivors and their adult children. The Holocaust survivor population in the United States gave Yehuda a research opportunity that was both scientifically significant and ethically demanding. These were people who had survived one of the most systematically brutal events in modern history. Many had lost entire families. Many had spent years in conditions designed to eliminate them physically and psychologically. And many had gone on to have children in America or Israel. Children who had grown up in relative safety, but who, clinicians had long observed, showed higher than expected rates of anxiety disorders, depression, and stress-related health problems. The clinical observation was not new. Therapists working with these families had noticed it for decades. What Yehuda brought to it was biology. Her team measured cortisol levels in Holocaust survivors and in their adult children, and compared those measurements to a control group of Jewish adults of similar age and background whose parents had not been in Europe during the war. Cortisol is the body's primary stress hormone. In people with post-traumatic stress disorder, cortisol regulation is often disrupted in a specific pattern, running lower than normal at baseline, reflecting a stress response system that has been altered by sustained exposure to extreme threat. What Yehuda found in the survivors was consistent with that pattern. What was less expected was what she found in their children. The adult children of Holocaust survivors showed altered cortisol profiles compared to the control group, even though they had not themselves been exposed to the events that had shaped their parents. Their stress response systems showed a biological configuration that resembled in measurable ways the configuration found in people who had experienced severe trauma directly. Yehuda and her colleagues then looked at the epigenetics. They focused on a gene called FKBP5, which plays a role in regulating the stress response and has been associated with post-traumatic stress disorder in previous research. In a study published in 2015 in the journal Biological Psychiatry, they found that Holocaust survivors and their children showed similar patterns of methylation at specific sites on the FKBP5 gene, and that those patterns differed from the control group in ways that were statistically significant. Before going further, it is important to be precise about what this finding does and does not establish. Yehuda herself has been consistent and careful about this in every forum where she has discussed the work. The sample sizes in these studies are small, a constraint imposed by the difficulty of finding participants who meet specific criteria and are willing to undergo the demands of the research. Small sample sizes mean that findings need to be replicated in larger groups before they can be treated as established. She describes the research as hypothesis generating, as pointing toward a question worth investigating further, not as providing a definitive answer. The study also cannot rule out non-biological pathways of transmission. Children of Holocaust survivors grew up in households shaped by their parents' trauma. The way those parents spoke, or did not speak, about what had happened, the particular silences that organized family life, the hypervigilance that many survivors carried into their parenting in ways that were often invisible to everyone involved. These psychological and social transmission pathways are real and well documented. A study of the size and design that Yehuda's team conducted cannot fully untangle the biological signal from those environmental factors. What the research does establish, within those limitations, is that there is a measurable biological difference in the stress response systems of Holocaust survivors' children that correlates with the epigenetic patterns found in the survivors themselves. Whether that correlation reflects biological transmission, shared environmental influences, or some combination of both, is a question the existing evidence cannot yet definitively answer. A second line of research approaches the same question from a different direction. On the morning of September 11, 2001, a number of women who were pregnant were near the World Trade Center in Lower Manhattan when the attacks occurred. Some developed post-traumatic stress disorder in the weeks and months that followed. Their pregnancies continued. Their children were born into a city still trying to understand what had happened to it. Yehuda and her colleagues studied a group of these women and their children. The findings, published in 2005 in the journal CNS Spectrums, showed that children of mothers who had developed post-traumatic stress disorder following the attacks showed lower baseline cortisol levels than children of mothers who had been similarly present but had not developed the disorder. The difference was present in infants as young as 9 months old. Before any explanation involving conscious learning or modeled behavior could carry much weight, the same limitations apply. Small sample. Confounding variables difficult to fully control. The finding points in a direction, not to a destination, but the direction is consistent. Across the Overcolics records, across the Dutch hunger winter methylation data, across the Holocaust survivor, cortisol and epigenetic findings, across the September 11th infant data, the evidence keeps returning to the same observation, approached from different angles, using different methods, in different populations separated by oceans and decades. The biological environment a developing body forms in appears to shape the stress response configuration that the child will carry. How far that influence extends beyond the first generation, how specific the transmission is, how large the effects are in the context of everything else that shapes a human life. These questions remain open. What the research has established is more modest than the popular versions of this story suggest, and more significant than a dismissive reading would allow. Severe stress, experienced during critical periods of development, leaves measurable biological marks. Some evidence suggests those marks may influence the biology of the next generation. The people who carry those marks did not choose them. And the question of what it means to begin life with a stress response system already shaped by a history you never lived through is one that the science is only beginning to find the tools to answer. Chapter 5. Fear Without a Teacher. There is a specific experiment that developmental psychologists have run, in various forms, for several decades. You take an infant, old enough to move independently, young enough that their experience of the world is still very limited. You place them on a surface called the visual cliff, a table with a thick glass top extending beyond the solid platform beneath, so that the infant can see what appears to be a significant drop, even though the glass itself is solid and safe. Most infants will not cross onto the glass section. They stop at the edge. They look down. Something in the body says no before the mind has any framework for evaluating what it is seeing. The infant has not fallen, has not been warned. Nobody in their short lives has explained that heights are dangerous. And yet the response is there. Eleanor Gibson and Richard Walk documented this in research published in 1960, and the basic finding has been replicated and extended many times since. Human infants show a wariness of apparent drop-offs that appears before they have any personal experience that would justify it. The same pattern appears with other categories of threat. Infants and very young children show elevated attention and heightened responses to images of snakes and spiders at ages when they have had no opportunity to learn through personal experience that these animals can be dangerous. The psychologists Arne Oman and Susan Minecka, in research conducted through the 1990s and early 2000s, documented that human beings are primed to associate fear with snakes and spiders far more readily than with objects that are statistically more dangerous in the modern world, such as cars or electrical outlets. A child who touches a hot stove learns quickly and specifically to avoid hot stoves. The learning is precise. It does not generalize beyond its source. But the response to a spider arrives already prepared, already leaning toward fear, before any personal lesson has been delivered. This is what the psychologist Martin Seligman described as preparedness. The idea that evolution has shaped human nervous systems to be differentially ready to associate fear with specific categories of stimuli. Not all stimuli are equal. Some arrive preloaded with a readiness to become threatening that others do not have. This is a real and well-supported phenomenon. And it is important to be precise about what kind of mechanism it represents, because it is not the same as the epigenetic inheritance described in the previous chapters. Preparedness is the product of genetic selection operating over vast timescales. The differential readiness to fear snakes is encoded in the architecture of the human nervous system through the same process that encodes every other evolved trait. Genes that produced nervous systems quick to respond to snake-like stimuli were passed on more reliably than genes that did not, over hundreds of thousands of generations. That differential survival shaped the nervous systems of every human being alive today. This is inheritance, but it operates through genetic selection across evolutionary time, not through epigenetic marks produced by the specific experience of a parent or grandparent. The timescales are different. The mechanisms are different. The level of certainty in the evidence is different. Preparedness explains why every human infant pauses at the visual cliff. It does not explain why the child of a Holocaust survivor shows a specific cortisol profile that differs from control groups in ways that correlate with the parent's trauma history. Those are different questions. They require different answers. And one of the persistent problems in popular accounts of this field is that the two are treated as the same thing, as if the fear of snakes and the cortisol profile of a trauma survivor's child are simply two expressions of a single phenomenon called genetic memory. They are not. Conflating them produces a story that is more dramatic than the science supports and less interesting than the actual picture. What preparedness does offer is something worth holding separately. It shows that the human nervous system arrives in the world already configured by history. Not personal history. Not family history, in the specific sense the epigenetic research explores. The deep history of a species that spent most of its existence in an environment very different from the one most people in the developed world now inhabit. The infant who stops at the visual cliff is responding to something real. The ancestral environment was full of drop-offs that killed people who did not respond to them. A nervous system quick to recognize that danger survived more often than one that did not. And that pattern of survival, repeated across a span of time, so long it cannot be meaningfully imagined, shaped the nervous system that every human infant carries into the world today. That is inheritance. Ancient. Slow. Encoded across a time frame that makes the 3 generations of the Dutch hunger winter research look like a single breath. Both of these mechanisms are real. Both of them mean that the body you were born with already knew something about the world before your own experience began. What they leave open is the question of the middle ground. The territory between the evolutionary timescale of preparedness and the single-generation prenatal effects of the famine and trauma research. The question of whether specific experiences of specific people, living within historical time, within the span of years that families can actually remember, leave biological traces that influence the nervous systems of their descendants in ways that are more targeted, more personal, and more recent than the broad species-level configurations of evolved preparedness. That is the question the epigenetics research is trying to answer. And answering it requires looking inside a part of the genome that scientists spent decades dismissing before they understood what they were looking at. That is where the story goes next. Chapter 6. The Junk That Isn't Junk. For most of the second half of the 20th century, there was a term that appeared regularly in genetics, textbooks, and in the lectures of professors teaching the next generation of molecular biologists. The term was junk DNA. It referred to the roughly 98% of the human genome that did not appear to code for proteins. The 2% that did code for proteins carried the instructions for building the molecular machinery of living cells. That part was underlunkitoed. It had a clear function. The other 98% seemed to do nothing at all. It was there, copied faithfully every time a cell divided, present in every living human body, and producing no apparent output that researchers could identify or measure. The term was not meant as a casual dismissal. Susumu Ono, the geneticist who popularized it in a paper published in 1972, was making a reasonable inference from the data he had. If a sequence does not code for a protein, and if coding for proteins is what genes do, then a sequence that does not code for proteins is, in the relevant sense, not doing anything. The problem was that the inference rested on an assumption that turned out to be incomplete. Coding for proteins is not the only thing that DNA sequences can do. The realization came gradually, through the accumulation of findings that did not fit the model. Sequences in the supposedly junk regions that were conserved across species, meaning that evolution had preserved them unchanged across millions of years. That is exactly what you would not expect of sequences that did nothing. Useless sequences accumulate mutations freely. Useful sequences are protected from change by selection. The conservation of these non-coding regions was a signal that they were doing something, even if what they were doing was not yet understood. Then, in 2012, a large international research consortium published the results of a project called ENCODE, which stood for Encyclopedia of DNA Elements. The project had spent years systematically mapping every region of the human genome that showed any measurable biochemical activity. The initial results claimed that roughly 80% of the genome showed some form of biochemical activity. That claim was immediately and seriously contested. Researchers including Dan Grauer at the University of Houston argued forcefully that biochemical activity is not the same as biological function. That just because a sequence is being transcribed does not mean the transcript is doing anything meaningful for the organism. The debate about how to interpret the ENCODE results continues in the scientific literature. And the original 80% figure is widely regarded as an overstatement, even by researchers broadly sympathetic to the project's goals. But the core findings survived the criticism. The genome is considerably more active, more complex, and more interconnected than the junk DNA model had suggested. The non-coding regions are not silent. They contain thousands of regulatory sequences, stretches of DNA that do not code for proteins, but that influence which protein-coding genes are switched on or off. At what times? In which cell types? In response to which signals? The orchestra needed not just musicians, but a conductor. And the conducting was happening in the regions that had been dismissed as useless. Within this revised picture, one category of sequence is worth pausing on: transposable elements, sometimes called jumping genes, are sequences that have the ability to copy themselves and insert those copies at new locations within the genome. They make up somewhere between 40 and 50% of the human genome by current estimates, though many are ancient copies that lost the ability to move long ago and now sit fixed in place. Remnants of molecular activity from millions of years back. The jumping genes were discovered by Barbara McClintock, working with corn plants in the 1940s and 1950s. She observed that certain genetic elements could change position within the genome and that their movement influenced the expression of nearby genes. Her findings were initially met with skepticism. She received the Nobel Prize in Physiology or Medicine in 1983, more than 30 years after her original work was published. When the broader scientific community had finally caught up with what she had observed, in the human brain, some transposable elements remain active. They are still inserting copies of themselves at new locations in the genomes of individual brain cells, contributing to genetic variation between neurons in a single brain. What role this activity plays in brain function, in learning, in the disorders that affect the brain, is a question researchers are actively investigating. These are early findings. They appear in peer-reviewed literature from reputable institutions, but they represent the frontier of current understanding rather than settled conclusions. What the story of the genome's hidden complexity adds to this broader investigation is a specific kind of perspective. For decades, scientists looked at 98% of the human genome and called it useless because they did not understand what it was doing. The tools they had were not precise enough to reveal the activity that was there. The framework they were working within was not capacious enough to accommodate functions that did not fit the protein coding model. They were not wrong to work within the best available framework. That is what science does. But the revision that followed is worth keeping in mind as this story continues. The absence of an understood function is not the same as the absence of a function. What looked like silence was, in many cases, a conversation happening at a level the available instruments could not yet hear. The genome is not a simple text with a small, meaningful section surrounded by filler. It is a regulatory system of extraordinary complexity in which the relationships between sequences, the way one region influences the behavior of another, the way environmental signals are translated into changes in gene expression, are still being mapped with tools that did not exist 20 years ago. Within that system, the epigenetic marks described in the previous chapters are not isolated curiosities. They are part of an architecture that is responsive to experience, that carries traces of history in molecular form, and that operates almost entirely below the threshold of conscious awareness, what that architecture is capable of transmitting across generations. Under what conditions, and with what consequences for the people who inherit it, is the question the research is working toward. The answer is not yet complete, but the question can now be asked with instruments precise enough to begin producing real data. And the data, as the next chapter will describe, has produced as much productive disagreement as it has produced consensus, which is exactly what you would expect from a question this large. Asked this early in the process of finding out. Chapter 7. The experiment that divided scientists. Science is not a collection of facts. It is a process, a method for asking questions in ways that allow the answers to be checked, challenged, revised, and eventually, if the evidence is strong enough and consistent enough and reproducible enough, accepted as the best available account of how something works. That process is slow. It is contentious. It is full of dead ends and premature conclusions and findings that seemed solid until someone tried to replicate them and found that the solidity was less reliable than it appeared. And it is. When it is working, as it should. Self-correcting in a way that no other method for understanding the world has managed to be. The science of transgenerational epigenetic inheritance is in the middle of that process right now. Not at the beginning, where everything is speculation. Not at the end, where the question has been settled and the debate has moved on. In the middle, where the findings are real enough to take seriously and contested enough that serious researchers disagree sharply about what they mean. Begin with the challenges, because they are substantial and they come from researchers whose credentials are not in question. Jerry Coyne is an emeritus professor of ecology and evolution at the University of Chicago. One of the most respected evolutionary biologists of his generation, he has been a consistent and direct critic of what he regards as overclaiming in the epigenetics literature. His position, stated clearly in published writing and in public commentary, is that the evidence for robust transgenerational epigenetic inheritance in mammals, and particularly in humans, has been systematically overstated. By researchers excited about a novel phenomenon, by journalists excited about a compelling story, and by a cultural appetite for findings that seem to confirm something people already feel to be true about the weight of family history. His specific criticisms focus on replication. The mouse study by Dias and Ressler has not been cleanly reproduced. Other researchers attempting to replicate the key findings have encountered difficulties. In a field where the headline result is extraordinary. Inconsistent replication is not a minor technical issue. It is the central scientific question. Other researchers have raised concerns about the mechanisms proposed to explain how epigenetic marks could survive the erasure that occurs during reproduction. The near-complete resetting of the epigenome during the formation of germ cells and during early embryonic development is an observed biological process. Not a theoretical assumption. The claim that some marks survive that resetting in ways that carry meaningful biological information requires specific mechanistic evidence. Critics argue that the evidence provided so far has not been sufficient to establish the proposed mechanism convincingly. There is also a concern specific to the human studies. The Holocaust survivor research, the Dutch hunger winter follow-up work, the September 11th infant data, all involve populations where the biological signal, if it exists, is entangled with social and psychological factors that are extremely difficult to separate. A child raised by a parent with post-traumatic stress disorder grows up inside a specific emotional world, a specific quality of attention in the household, a specific set of unspoken lessons about what the world is like and what it is capable of. Separating the biological transmission of epigenetic marks from the social transmission of that emotional world requires a study design that none of the existing human research has been able to achieve. Yehuda acknowledges this directly and consistently. She describes her work as hypothesis generating. That honest positioning reflects the actual state of the evidence, rather than the state of the headlines the research generates. It is worth noting, every time her name appears in this story. Now consider what the evidence does support. Transgenerational epigenetic inheritance is well established in plants. The mechanisms are understood. The effects are robust and reproducible. In the roundworm known as C. elegans, a small organism that has been one of the most important subjects in genetics research for decades, the phenomenon has been demonstrated convincingly, and the mechanisms have been worked out in significant detail. In Miche, the evidence is more contested, but not absent. Some effects have been reproduced. The mechanisms remain under investigation. In humans, the evidence is at its most preliminary. The Dutch hunger winter methylation findings in the directly exposed generation are robust. The evidence for transmission to subsequent generations is present but not established with the rigor that would allow confident conclusions. This produces a picture with three distinct layers. The first layer is confirmed. Epigenetic marks exist, respond to environmental signals, and influence gene expression in ways that affect health and behavior throughout an individual life. This is established science, not seriously contested by anyone working in the field. The second layer is strongly suggested but not yet confirmed. Some epigenetic marks can survive the reproductive erasure process and influence the biology of the next generation, at least in some organisms under some conditions. The evidence for this in mammals is present and taken seriously, but faces replication challenges that prevent confident conclusions. The third layer is speculative. Transgenerational epigenetic inheritance in humans is robust enough and specific enough to meaningfully transmit information about ancestral experience across multiple generations in ways that substantially shape behavior and health. This is an interesting hypothesis supported by suggestive data. It is not an established finding. The distance between the second and third layers is where most of the scientific controversy lives. And it is the distance that popular accounts of this field most consistently collapse, treating the speculative layer as if it were as well supported as the confirmed layer, and presenting the confirmed layer as if it were evidence for the speculative claims. It is not. Hold that distinction clearly, not as a disappointment, as a description of where the science actually is, because the actual picture, held at the level of accuracy the evidence supports, is genuinely remarkable. It does not need inflation to be worth attention. The jury is still deliberating. That is not a failure of the science. It is the science working exactly as it should. The failure would be to announce a verdict before the deliberation is complete. In either direction, to declare the phenomenon proven when the evidence does not support that conclusion, or to dismiss it entirely when the questions it raises are genuine and the investigation is ongoing. The most honest thing this story can offer is the deliberation itself, held openly, reported accurately, without reaching for more than the evidence will support. That honesty is not a limitation. It is the most interesting thing about the story. Chapter 8. What other knowledge systems observed? There is a particular kind of knowledge that does not begin in a laboratory. It begins in observation, in the accumulated attention of people watching, across generations, how human beings behave, how families carry patterns forward, how the particular quality of a wound in one era appears, transformed but recognizable, in the lives of people born long after the original wound was inflicted. It is knowledge built not through controlled experiments, but through the kind of long, careful, multi-generational attention that only becomes possible when a community stays together long enough and pays close enough attention to notice what repeats. This kind of knowledge exists in many traditions around the world. It predates the laboratory by thousands of years. And as the science of epigenetics has begun asking questions about what biology carries across generations, researchers in anthropology and the history of science have noted something worth recording carefully. Several knowledge systems, developed independently in different parts of the world, arrived at observations that bear a structural resemblance to what the epigenetics research is now attempting to measure. This requires precise framing before going further. What follows is not an argument that traditional knowledge systems predicted epigenetics. It is not a claim that ancient wisdom confirmed what modern science is only now discovering. Those framings are inaccurate and they do not serve honest understanding. Modern biology is not proving those traditions right in a simple way, but it is beginning to ask questions that make the comparison worth handling carefully. With that framing in place, consider what those traditions actually observed. Among several indigenous nations of North America, there exists a concept that anthropologists and cultural scholars have documented under various names, but that is most commonly translated into English as blood memory. As recorded in ethnographic literature and in published interviews with community members, the concept refers to something carried in the body rather than in the conscious mind, a knowledge or orientation or sensitivity that passes through lineage without being explicitly taught, something a person may discover in themselves, a response to a place, a recognition, a particular quality of attention towards certain kinds of experience that seems to come from before their own life began. The concept is not uniform across all nations or communities that use the term. It carries different specific meanings in different contexts. It is embedded in cosmological frameworks that differ significantly from one tradition to another. Any account that flattens those differences misrepresents the actual complexity of the traditions involved. What is consistent across the various uses of the concept is the observation that lineage transmits something that is not reducible to explicit teaching, that the body carries a relationship to history that the conscious mind did not acquire through personal experience. That observation, made within a specific cultural framework and carrying specific cultural meanings, is structurally similar to the question that the epigenetics researchers have been asking in their laboratories. Not identical, not interchangeable, structurally similar. In the Jewish tradition, the concept of zikaron, which translates most directly as remembrance, carries a weight that extends beyond individual recollection. The instruction to remember, repeated throughout the Hebrew Bible and elaborated in rabbinic literature, is not simply an instruction to keep a historical event in mind. It is an instruction to carry that event in the body, to allow it to shape present behavior and present identity. The Passover Seder instructs participants not that their ancestors were slaves in Egypt, but that they themselves were slaves. The memory is meant to be inhabited, not merely observed from a distance. Rachel Yehuda, who is herself Jewish and who has reflected in interviews on the relationship between her cultural background and her scientific questions, has noted that her tradition gave her a particular sensitivity to certain questions she might not otherwise have thought to ask. The tradition did not provide the answer. It oriented her attention toward the question. That is perhaps the most accurate way to understand the relationship between these knowledge systems and the epigenetics research. They did not predict the science. They cultivated attention toward a class of phenomena that the science is now beginning to measure. The attention was real. The observations were genuine. The frameworks within which those observations were interpreted carry meanings and implications that do not translate directly into molecular biology and should not be forced. But the fact that multiple knowledge systems, developed independently across different cultures and different centuries, arrived at the observation that what happens to people is carried forward in their descendants in ways that exceed explicit teaching, is worth noting carefully. It suggests that what the epigenetics laboratories are now working to measure with methylation assays and cortisol profiles is not so subtle or so hidden that only modern instruments could have noticed it. It is visible to careful observers at the level of human families moving through time. What the laboratory adds is not the observation. The observation was already there. What the laboratory adds is the ability to look inside the cell and ask whether a mechanism exists that could account for what those observers noticed. To try to find, in the molecular detail of living biology, a process that might correspond to the patterns that careful human attention had been recording in a different language for a very long time. It is not a mystical claim in this context. It is a biological question, one that the science has not yet fully answered, but one that it is now, for the first time, equipped to seriously ask. Chapter 9. You are not just you. Consider what it would mean to take seriously, even provisionally, the picture that the previous chapters have been assembling, not the speculative edges of it, not the claims that have not yet been established, the confirmed center of it, and the strongly suggested territory just beyond that center, held carefully and without inflation. It would mean this. The cells of your body contain epigenetic marks, molecular annotations on your genome, that were shaped not only by your own experience, but by the environment your mother's body moved through while you were forming inside it, the stress she experienced, the nutrition she had access to, the degree of safety or threat that organized her nervous system during the months of your development. It would mean that some of those marks may carry traces of environments that your grandparents moved through, though the evidence for this in humans is still being carefully examined and should be held with appropriate uncertainty. It would mean that the configuration of your stress response system, the particular thresholds at which your body moves from calm to alert, the specific sensitivities your nervous system carries, are not purely the product of what you have personally lived through. They reflect, at least in part, a biological inheritance that began before you did. That is a careful claim. It is the claim the current evidence supports. And even at that careful level, it changes something. Because it means that the body you are moving through the world in right now may not be a blank slate that your own experience wrote upon from the beginning. It may have arrived already shaped, already oriented, already carrying, in molecular form, some of the history of the people whose biology eventually produced yours. Not their memories, not their stories, something more quiet than that, and, in some ways, more intimate. A biological configuration. A set of molecular adjustments shaped by what those people survived and how their bodies adapted to survive it. Think of the woman in the Dutch city in the winter of 1944. The one who was pregnant and cold and eating tulip bulbs to stay alive. She did not know, could not have known, that researchers would one day find a specific methylation difference in the cells of the child she was carrying. She was only trying to get through the winter. She was only trying to keep them both alive. And she did. And something of that effort, something of the biological configuration her body produced under that pressure, passed forward, not as a story. She may never have told the story at all. Many survivors did not. But in the cells of the child who survived that winter inside her, something of what she endured may have left a mark that her own words never needed to carry. That is what the research suggests. Carefully, within its limits, with all the appropriate uncertainty attached, the philosopher Derek Parfit devoted much of his published work to the question of where one person ends and another begins. He did not engage directly with epigenetics research in his writing, but the questions he asked about the edges of personal identity point toward the same territory. This research is beginning to map from a different direction. The boundaries of the self are less sharp than they appear. Not in a mystical sense, in a biological one. The epigenetic marks in your cells may not have begun entirely with you. The stress response thresholds your nervous system uses did not emerge fully formed from your personal history alone. The body you inhabit is something like what manuscript scholars call a palimpsest, a surface on which multiple layers are present simultaneously. The most recent, visible on top, older layers still there beneath it, still influencing the surface, above them, even when they cannot be directly read. What this means practically deserves careful thought. On one hand, it offers a possible reframing of experiences that have long resisted explanation. The person who carries an anxiety their own life does not seem to justify. The sensitivity that seems to come from nowhere and yet feels deeply familiar, as if it belongs to something older than the individual life that contains it. These experiences are real. They have always been real. What the epigenetics research adds, tentatively, and, with the caveats, the evidence requires, is a biological dimension to the question of where they come from. Understanding that a stress response system may be calibrated in part by environments that preceded your own experience does not dissolve the difficulty of living with that calibration. But it may change its meaning. What seemed to be an unexplained irregularity can be understood differently when it is placed in a lineage. Not as something wrong with the individual who carries it, but as a biological inheritance. A configuration that made sense in the environment that shaped it. Even if it does not serve the same function in the environment the current person actually inhabits. That reframing is not a cure. It is a map, and maps, even incomplete ones, are more useful than the absence of maps. On the other hand, the picture assembled by this research points in a more uncomfortable direction as well. If what you carry in your cells may include some biological trace of the environments your ancestors moved through, then what you pass on will include some biological trace of yours, the stress response your own history has shaped, the epigenetic configuration that your life has produced, carried forward into cells that will become people who will move through a world you cannot predict. That is not a reason for guilt, but it is a reason for attention. The research on prenatal stress, the September 11th cortisol findings, the Dutch hunger winter data, all of it points toward the same observation. The environment a developing body forms and shapes the biology it will carry, not irreversibly. The epigenome is responsive to experience throughout life. The configurations laid down early can be modified by subsequent experience, by relationship, by the quality of care and safety that a life provides. But the starting point is not nothing. It is a biological inheritance that reflects history. What you do with that starting point is not predetermined. There is one more thing worth saying in this chapter. If the research is right, even in its most careful and limited form, then the people who came before you did not vanish entirely when their lives ended. Some biological trace of the environments they moved through may be reflected in the cells of the people who came after them. Not as memory, not as presence, as chemistry, as molecular adjustments that their lives contributed to a chain that eventually included yours. The woman in the Dutch winter, the man who survived something he never spoke about, the grandmother whose particular quality of alertness you may have always recognized in yourself without knowing where it came from. Some trace of what they endured may be present in you in a form that is real and measurable and entirely below the threshold of anything you could consciously access. That is a tentative claim. The science that points toward it is preliminary and contested and should be held as such, but it is worth sitting with quietly. Not as a certainty, as a question worth taking seriously. Because if it is even partially true, it changes something about what you are. Not dramatically, not in ways the thinking mind can fully process or the language can fully capture, but in the way that quiet things change you, the way a piece of information, placed in the right context at the right moment, rearranges something that had always been there but had never been seen from that particular angle before. You may not be only yourself. You may also be, in some small and still being measured biological sense, a continuation of the people who made you possible. Their lives shaped something that reached forward into yours. And in the quiet of this particular moment, with the day behind you and the thinking beginning to slow, that may be less unsettling than it first appears. Because it means that you are not alone in the way that a single life, moving through a single span of years, might seem to be alone. You carry something forward, as they did, as everybody in this long chain has done, without knowing it, without being asked, simply by surviving long enough for the next one to begin. Chapter 10. What the Science Can and Cannot Say. There is a particular kind of courage that honest science requires. Not the courage of the explorer pushing into unmapped territory, though that quality is present too. The courage of stopping, of looking at what the evidence actually shows and what it does not show, and being willing to draw the line between those two things in a place that is accurate rather than satisfying. That line is harder to draw than it sounds. Because the findings in this field are genuinely interesting, the questions they raise are genuinely important, and the temptation for researchers and for the people who communicate their work to broader audiences is to let the interest of the questions pull the conclusions further than the evidence will honestly support. This story has tried not to do that. This chapter exists to check that work. Begin with what is confirmed, because the foundation matters before anything built on top of it. Epigenetic marks exist and influence gene expression in real and well-documented ways. The process of methylation, by which chemical groups attach to specific sites on DNA and alter how genes in those regions are expressed, is established molecular biology. The responsiveness of those marks to environmental signals, including stress, nutrition, and chemical exposure, is documented across a substantial body of peer-reviewed research. This is not contested. It is the solid ground beneath everything else in this story. The effect of prenatal environment on the epigenome of the developing organism is established. The Dutch hunger winter research demonstrated convincingly that severe nutritional deprivation during fetal development produces measurable methylation differences, particularly at the IGF-2 gene, that persist for decades in the directly exposed generation. This finding has been replicated. It is not seriously contested by anyone working in the field. The general principle that early environmental conditions shape the configuration of stress response systems in ways that influence lifelong health is established. The mechanisms through which prenatal and early postnatal stress influence cortisol regulation, immune function, and metabolic processes are documented and represent mainstream findings in developmental biology. These confirmed findings are significant. They establish that the body you inhabit was shaped, in measurable ways, by the environment you were formed in. That is a genuine revision of the picture of human development that dominated biological thinking for most of the 20th century. In some important respects. Now, move to what is strongly suggested but not yet confirmed. The transmission of some epigenetic marks from parent to offspring, surviving the reproductive erasure process in at least some organisms under at least some conditions, is strongly suggested by the available evidence. In plants, and in the roundworm known as C. elegans, this transmission is established and the mechanisms are understood. In Miche, the evidence is present and taken seriously, but has faced replication challenges that prevent confident conclusions. In humans, the evidence for transmission of marks associated with parental experience to the next generation is present in the literature. The Dutch hunger winter studies showing some methylation differences in children of famine-exposed mothers. The Yehuda research on FKBP, 5-methylation patterns and cortisol differences in children of Holocaust survivors. The September 11th infant cortisol findings, all pointing in the same direction, all doing so with modest sample sizes. Difficult to control confounding variables, and effect sizes that are real but not large. The researchers responsible for these findings describe them consistently as preliminary. That description should be taken at face value. Now move to what is speculative. The claim that transgenerational epigenetic inheritance in humans is robust enough and specific enough to meaningfully transmit information about ancestral experience across multiple generations, substantially shaping the behavior and health of people living several generations after the original experience, is speculative. It is an interesting hypothesis supported by suggestive data. It is not supported by evidence of the quality and consistency that would allow confident conclusions. That is where the line is. Confirmed at the base, strongly suggested in the middle, speculative at the top, and the honest position is to hold each layer at its own level rather than allowing the interest of the question to pull all three into the same category. What the science is now equipped to do, in ways that were not possible a generation ago, is ask these questions with tools precise enough to produce real answers. Single-cell sequencing allows researchers to examine epigenetic marks at a resolution that was unimaginable 20 years ago. Multi-generational cohort studies with biological sampling at multiple time points are generating datasets large enough to begin separating signal from noise in human populations. Computational methods are being applied to the complexity of epigenetic data in ways that earlier analyses could not attempt. The picture that emerges from these tools over the next decade will be more detailed and more reliable than anything currently available. Specific questions that remain open today: about which marks survive the reproductive erasure process in humans and under what conditions, about the size and specificity of any transgenerational effects, about the mechanisms through which prenatal stress influences offspring biology. We'll have better answers. What those answers will show is not yet known. But the direction of the current evidence, held without inflation, points towards something real, something that adds a layer to the picture of human inheritance that the standard genetic model did not have room for, something that connects individual lives with their particular histories and their particular biologies, to lineages that extend further back than personal memory and further forward than any individual lifespan. What that connection means for how you understand yourself and the people who made you possible and the people you may make possible in turn is a question the science can inform but cannot answer. It belongs to the quiet, to the particular quality of attention that a night like this one makes available, which is exactly where this story has been heading all along. Chapter 11. Closing. Let your breathing slow a little more now. The hard work of following an argument is behind you. What remains is simpler. You have spent this time inside a question that does not have a clean answer. A question about what you carry, about where the body's knowledge comes from, about how far back the shaping of a single human life extends. The science has given you some of what it knows, clearly and without inflation, and it has been honest about what it does not yet know. That honesty is not a disappointment, it is an invitation. Somewhere tonight, a researcher is looking at methylation data from a multi-generational cohort study, trying to separate the signal from the noise in a dataset that required years to collect. Somewhere, a developmental biologist is watching the epigenome of an embryo in the earliest hours of its formation, trying to understand which marks survive the erasure process and why. Somewhere, a clinician is sitting across from someone whose nervous system carries a configuration that their own life does not fully explain, and is finding, slowly and carefully, a way to use that understanding, not as a verdict, but as a starting point. The investigation is ongoing. The questions are alive. And in this particular moment, in the quiet of wherever you are, with the thinking mind beginning to soften and the body beginning to do what bodies do in the dark when the day is done, something is worth noting. You are a continuation, not a beginning, not an isolated point in time assembled from nothing, a continuation of a line that extends back through more lives than any archive could hold, through people who survived things you will never know about, through people who loved in circumstances you cannot fully imagine, through people whose names have been lost so completely that not even the memory of their loss remains. The woman who was pregnant in the Dutch winter, the man who came home from something and never spoke about it, the grandmother who held her children with a particular kind of alertness that her own children recognized without knowing why. Some biological trace of what they endured may be present in the people who came after them. The science is careful about how much it claims. Preliminary. Contested. Still being worked out in laboratories and cohort studies and sequencing runs that will not be published for years. But the direction the evidence points is toward connection rather than isolation, toward a picture of human inheritance that is more layered, more continuous, and more human than the standard model allowed for. You did not choose what you carry. Nobody does, but you carry it. And in carrying it forward, you are doing what every body in this long chain has done, without knowing it, without being asked, simply by surviving long enough for the next one to begin. So let the thinking go. Let the questions rest where they are. They will still be there in the morning, patient and open, waiting for whatever new instrument or new finding brings the next piece of the picture into focus. Tonight, you have done enough. The cells are doing their work, reading the marks, running the machinery, carrying something forward that nobody chose to keep and nobody has yet found a way to fully read. You are a continuation. Rest now. Good night.